All right, looks like we still have to use this, but the afternoon session will not be recorded, but still on the Zoom, I think. All right, brilliant. All right, thanks for coming back. And thank you, Stefanie. So we juggled the schedule a little bit, and to accommodate Otto, who's going to be speaking tomorrow now at 11, and Sunil, who's supposed to speak at 11, will speak in the morning, and then Stefanie, who's supposed to speak tomorrow morning, kindly agreed to speak now. So, you know, sort of extra special claps at the end for Stefanie for helping us. And also I should mention, Stefanie helped us a lot during the first version of this meeting in 2014. And actually on that paper that I mentioned, she was the first author and the main driver of that paper. So thanks a lot and great having you again, Stefanie.
Thank you. Can you hear me? Is the mic on? Okay, so my name is Stefanie Widder and I'm a computational systems biologist, and I'm currently at the Medical University in Vienna. And my main research focus is on trying to understand how microbial interactions drive emergent properties at the community level. And today I will be talking a lot about community structure. And I wanted to clarify what I mean by this. I mean the topology of co-occurrence networks. In other words, the dependencies and competitions between organisms in the community. And also I wanted to point out that this is all recent work, so it's unpublished, so please don't overly share. Let's put it like this. My focus is on cystic fibrosis. It's the lung microbiome in cystic fibrosis I'm looking at. And cystic fibrosis is a genetically born disease. People have a mutation in the CFTR gene. This is a transporter that is expressed on all the mucosas of the body. And I'm looking at the lung because there, this deficiency results in a too viscous mucus. And this mucus invites a permanent colonization, a polymicrobial infection that is persisting over a lifetime of the patient. And this lung microbiome has all hallmarks of a clear complex system. It has a spatial heterogeneity in terms of where it's actually, which part of the lung you look at. It has temporal succession throughout the patient's lifetime, and it has a very particular environment that depends on the treatment history of the patient. And then you have many different organisms that are kind of living together or competing in this microbiome, including not only the traditional CF pathogens, but also anaerobes, for example, in the lung. Yeah? And the disease itself manifests in iterative so-called pulmonary exacerbations. This is an overshooting immune response that results in a lot of scarring that is irreversible. And so this means that over time, the more often you have this exacerbation, the quicker your lung function declines and the quicker you have a premature death event. And basically the major objective of the field is to somehow predict these exacerbations in order to avoid them. Yeah, and our hypothesis on this was that there should be some changes happening before this overshooting that can actually be detected in a way, some disequilibrium in the microbiome state that is then directly related with the inflammatory state. But having said this, it was a very difficult task to actually come up with. And the reason for this is that we have, so to say, processes that are strongly overshadowing any small-scale signals that we could be looking for. The first one is a very strong patient specificity. Yeah? And you see here an ordination, a fate ordination, which takes into account time series. So on X and Y, you have the ordination and T of time. And these are roughly 9,000 samples, and they're colored according to patient. And what you can see is that these individual time series stay in their individual proportion of the ordination and never ever, change or intermingle. So it is very difficult to find a way of how to group these individuals. Yeah? And if you look at the time series, this is an individual patient, and here you see the composition of 4 groups. So we grouped the organisms in 4 groups. These are traditional CF pathogens, anaerobes, aerobes, and facultative anaerobes. And you can see that, I don't know if you see it very well, but you can see that there is kind of overall the same pattern happening, and then sporadically you see some flares. And then if you look, and this is the treatment, the acute treatment that people are getting, these flares are associated with the acute treatment. You can also see this if you look at the brachyartery distance of the samples. But later on, basically everything comes back to how it was before, more or less. Not at every organismic detail, but in these large groups, there's a lot of resilience. And we have these treatment biases in the data. So on top of this, we also had clinical evidence that there is not one type of exacerbation going on. So looking for one thing and the commonalities between patients for one thing, but could be more different things, more different processes that are actually going on. And so in this project that I will be talking today about, we tried to use network science and modeling to overcome this patient specificity in any way, and also reduce a little bit the treatment bias. We cannot get rid of it in total, but at least reduce it a little bit. And if this is possible, then the idea would be we can actually group patients that have more commonalities or look more alike in their trajectories, and find perhaps some exacerbation regimes that we can then characterize by their community structure, by the ecological processes that are going on. And perhaps even finally propose something in terms of drug targets or a proposition on how to best treat. Okay, and this is what I will be showing you. So the first part is actually for us for a long time the most important and the most difficult part, how to overcome all these confounders that I've been just talking about. Yeah, and finally, we came up with the following strategy. So I didn't talk about the data that I'm having. So we started with a data set of 9,000 samples. These were sputum samples, and these are 16S sequenced. And from these 9,000 samples, we thought it would be a good idea to have or extract a subset to something that could be a prototypical trajectory, which means we wouldn't want to have the acute treatment samples in there, of course, in order to reduce this. And also we wanted to have like a reasonable time length. So we decided we take 60 samples before acute treatment, and wanted to have samples, so to say, distributed across these 60 days before treatment, sorry. So this is daily sampled actually, in most evenly. And so from these 9,000 samples, we actually in the end had 800 samples. So all the rest did not fit into this rigorous criteria. These were from 11 patients, and altogether we had 18 of these individual trajectories. Yeah? So this is the data we go in. And then we moved from looking at individual organisms to other kind of matrices that would be representative of this sample. And I think this was actually the key part. And I will not now explain how we did this, but if you want to know, you can ask me later. But however, we were looking at various aspects. We were looking at the ecology of this sample, at the community flavor, and some functional aspects. And with this, we had a different descriptor of the sample, and we could go on and go and ordinate. And this is in fact the ordination that we got, even though as a thumbnail. And you can see, as very opposed to the first ordination that I showed you, here we have some overlap between the samples, also again colored by a patient here. And this was the big breakthrough, if you want to. And then from that, you can do clustering and you can aggregate. And this is what here, this is the heat map that we had. Please note that on the x-axis, we don't have samples, but we have the principal components, yeah, first 3 principal components. And then we have some additional layers of information, Dirichlet multinomial mixtures. I will talk about them in a second. So this is a way to broadly define a community type, the patients and the exacerbation cycle, so to say, the individual trajectory. And you can see by the bare eye that there is a lot of order in that, yeah? And so we had to convince ourselves into how many clusters we want to actually cut this data set. No? And so we were using a chi-square test with different numbers of clusters on the x-axis, and we were just looking for the first flattening, so to say, and convinced ourselves that this could be perhaps perhaps the ideal way to cut this would be 3 clusters. This is debatable. Yeah. Okay. But now we had these 3 clusters, and all of a sudden we had more patients within one group that we could compare between. Yeah. And you can see here, these are the individual samples colored by these 3 clusters. This is one patient, another patient, and these are individual trajectories. And you can see that most of the time the samples associate with one cluster, only sporadic outliers, so to say, particularly for the first cluster. For the second 2, there is more mixture. Some of them are even very heavily mixed, and I removed those from the follow-up analysis. If you then compare patient ID to the clusters, because this is also a question, so how stable are the patients within these regimes? Then we could see again the same picture that you can also see just by looking at this plot. Cluster 1 defined very nicely, so you have 1 blue blob and 2 red blobs for all the patients that are clustering in this group. And cluster 2 and 3 sometimes was very nice, but sometimes had a little bit of an overlap. And you will see why this is the case in a second. So now we're in a position, we have groups, and let's now try and better characterize them. And we were looking at temporal dynamics within and between these groups, microbiome structure, and a little bit into community function. And I show you here again an overview. So the very first thing is just temporal behavior, where you have time to treatment on the x-axis, and you look at different properties and how they change. The second part will be about co-occurrence networks, and we heard this before. So we inferred co-occurrence networks in a sliding window. We used a certain number of samples, and we skipped by 1 individual sample. And this means that if I have 60 samples altogether, I have 40 inferred co-occurrence networks. And this is a large number. And from this large number, we can actually do network analysis and also statistical network analysis, and then get even some insight in the community roles, as you will see in a second. Okay, so the first thing in terms of time dynamics that I wanted to show you, here's a little mistake, is how the Dirichlet multinomial mixtures behave over time. And we have the principal developer of this kind of model here in the audience. The idea here is that you have a big data set and you find representations, a reduced number of representation where individual samples are then just instances of this community abundance distribution. Yeah? And from our data set, as I said, 800 different samples, we obtained 6 different mixtures. And here you see 3 plots. This is cluster 1, cluster 2, cluster 3. And you can see that the coloring is, so to say, coherent. You can see that we have a different coloring here than we have here. And in this case, we have communities that are dominated by the pathogens mostly. And here we have communities that are dominated by anaerobes. And since both of them are anaerobic, so this is why the delimitation of these 2 clusters is not working as well. Yeah? Okay, if you look at time behavior, there is no significant time behavior in terms of the broad community composition. But this is something that we would have expected anyway, also from the stability in the patients. However, if you look at diversity measures, so Shannon or Chow, so diversity or richness, and here you have the 3 clusters in the 3 colors, we see, and on the x-axis we have now days to treatment, so 0 days to treatment, 60 days to treatment, yeah, far away, very close to treatment. We can see that the first cluster is declining or increasing, but I'm reading it towards treatment, yeah. So the first cluster is declining, also decreasing Shannon diversity towards treatment, whereas cluster 2 and 3, the anaerobic ones, are increasing. And this is the same case for the Chow richness. All of these are significant with the exception of the cluster 2 for Chow, yeah? But cluster 3 for Chow. Cluster 2 is increasing, so the first anaerobic cluster is increasing, and the cluster 1, the pathogen cluster, is decreasing. Yeah. Okay. So by looking at this, and we will see that this is happening more often, so to say, we see that we have trends, we have significant trends, but they are not overly strong, right? They are weak trends. And you can imagine that if you don't have this ordering below everything, these would just cancel out, right? And this was one of the major problems in the field, that people would collect and collect and collect more, and there were just no consensus on what's actually happening, because these things cancel out, and then you don't know if you have this or that patients. Furthermore, we were looking at the turnover in the community. So now on the x-axis you have the Aichi-zone distance from one sample to the one yesterday, 2 days ago, and 20 days ago. And you would expect, of course, that a closed sample is more similar. So this is why this is increasing over time. And here I show again the 3 clusters, but in 2 different groups. Shortly before treatment, which is actually when the exacerbation should be happening. Yeah, so this is where we expect the signal building up, and further away from treatment. Yeah, and interestingly, you see, so red is shortly before treatment, blue is far away from treatment. Interestingly, you see again this antagonistic behavior. Yeah, in cluster 1, you see shortly before treatment decreased turnover. Further away, it is a little bit increased. And the other, in the anaerobic conditions, or in the anaerobic dominated communities, you see exactly the opposite. So shortly before treatment, you have an increased turnover, more organisms are changing, and further away, there is less. Again, this antagonistic aspect that you wouldn't catch otherwise. Okay, and now let's come to the network analysis. Yeah. I would like to summarize by now. So we have 3 different clusters. Basically, it seems like we have 2 different regimes. We have one that seems to have a stuck turnover, lesser diversity, and is dominated by pathogens. And we have the other one which is dominated by anaerobes, where there's a lot of turnover and a lot of diversity towards treatment. Yeah. Our idea by then was that we are looking at the dysbiosis and something else, and there will be more arguments that this is a strong dysbiosis in the first cluster. Okay, so I told you before, so we have 18 individual trajectories. I can infer very many co-occurrence networks in a sliding window, and hence I can then look at the topology and can make some statistical tests. And what you see here are the 3 clusters, again compared to each other. All the measures that I'm using are centralized. Yeah, so I'm looking at node prevalence, edge prevalence, and between centrality. We see lesser nodes, lesser edges, and the higher between centrality in this cluster 1, and the opposing effect in the anaerobes. I would like to say something about the between centrality, because I'm not sure how many of you are familiar with these terms. So for the between centrality, we're looking at the structure of the graph. The highest between centrality is a graph that is a star, yeah, where you have one node in the middle and everybody else is connected in a star. Yeah. In terms of the interpretation of why we see a stronger between centrality for all the nodes, so to say, for the overall graph in this cluster 1 is for me that there's a more direct communication structure than is in the other case from of the anaerobes. So we see smaller, and I forgot to say, so this is all done on the giant component. So it's not done on the entire network, yeah, but only on the giant component, because the giant component is the one that is the largest part of the graph that is connected to each other and most likely dynamically relevant, or most relevant as compared to other pieces of the graph. So smaller, simpler, more straightforward in communication. And then, sorry. Yeah. And then I was also looking at hierarchies in these networks. I fell over a hallmark paper from 2004, Barabashi Oldby. I actually teach this paper, and then when I was reteaching it, I was like, oh, why didn't I think of it before? And what they are proposing is basically that you can look at two different distributions, and it will tell you if you have a hierarchical network or not. And it comes actually from this idea of master gene regulator, and this is the typical or the prototypical hierarchical network where you have the master regulator in between. The master regulator will talk with the individual modules, but the modules themselves will not talk to each other. So this is the prototypical hierarchical system that you can kind of predict or represent with these two different distributions. And we're talking about the degree distribution, and we're talking about the clustering distribution. Okay, so the idea is you have to fit the power law, and if this power law is somehow fine, then you're good. So this is over so many different networks, so I was quite fine actually to have this kind of semi-power law. And I think perhaps the most important part here is now looking at the clustering distribution, looking at the slope of the linear fits. Because the slope of the cluster 1 is much steeper, as you can see here in the anaerobes. This suggests that we have a very strong hierarchy in these simpler, better communicating systems driven by pathogens, whereas this is not the case in the anaerobic system. So taken together, I think we have the hallmarks of a strong dysbiosis. We have drivers, we have a clear communication, we have hierarchy, and we have a reduced variability in reality. Yeah, okay. And then we were, of course, interested. Now that we had this idea of the hierarchies, we wanted to better understand who was actually sitting on top of these hierarchies. Yeah? So we have many different organisms, many different graphs. And the idea was then to pick for each graph individually the most hierarchical position possible. Yeah? So this means in the case of the cluster 1, this might be really a strong hierarchical position, whereas in the other clusters, it might be a more shallow hierarchy. But it is still within the graph the most hierarchical position. And then we were counting how many times the different ASVs were appearing on the most hierarchical positions. Yeah? And we got out 3 very different distributions, which I found was really nice. So for the cluster 1, the dysbiotic cluster, we find a distribution where everybody who is actually appearing is basically nearly appearing at the same rate. Yeah, so they are splitting up and very strong and sitting on top. And importantly, who is sitting on top? Pseudomonas and Staph and Strep. Pseudomonas is the canonical CF pathogen, and Staphylococcus as well, and Streptococcus. These are individual ASVs. I just didn't write the 1, 2, 3 here. So and Strep is as well. There is a single organism that's actually an emerging CF pathogen. So they are sitting, in fact, in these top hierarchical positions. Now, if you look at cluster 3, you get this broad distribution. Remember that the hierarchy was not as well pronounced. This is also reflected here because very many different organisms can sit in this more hierarchical position, and it seems to be not so conserved or not so relevant in the end. And one point that I would like to say as well is that even though we have Pseudomonas in these data, they not appear at any they don't appear to be in any hierarchical position in this different context. Okay, and so finally, we wanted to know a little bit more about the CF pathogens and whether we could find a way to describe their activity in the different clusters. And so one thing that I tried was to look at PageRank. Again, on the topologies, PageRank is like the eigencentrality, it's like the most important thing in the network. Yeah? And I split the count data into the 4 different groups: CF pathogens, aerobes, anaerobes, and facultative anaerobes. And then I was just comparing for the 3 different clusters whether there was some differences in the importance with regards to the other groups. And what you can see here, perhaps you don't see it very well, that in the dysbiosis, the CF pathogens are indeed more important. So not only they sit on top of the hierarchy, but they're also more important in terms of the topology, of the entire dynamic in the network. And the same grouping here, for example, where you look at the CF pathogens and all the others, you can see there is no difference in their activity and in their importance of inferred from the topology. Yeah. Okay. And then finally, we're also looking at noise colors. So this is a paper that we have been generating with Caroline and me some years ago, where we could show that the type of the underlying process can be, how can I say it? So if you imagine a time series and you do a noise analysis on this Fourier transform, you can get different colors of noise. And we could show that the underlying process, dependent on what kind of process it is, would produce different colors. Yeah, so a stochastic process obviously will produce white noise. But then the more deterministic you become, the darker the colors become. Yeah, and so in that sense, I was looking also on the noise that the CF pathogens versus the anaerobes would be emitting. And what I could see, and this again is here in these two groups, time groups, during exacerbation, if you want, shortly before treatment and far away from treatment. This is again the cluster 3. So in cluster 3, for example, the pathogens versus the anaerobes are basically producing white noise. So this is the stochastic behavior, and there's stochastic dynamics underlying this. Yeah. But if you compare it to our first cluster, you can see also a lot of white noise, but also a lot of pink noise, the darker noise, like suggesting there's more deterministic going on, more interactions, more straight behavior. And also in terms of during exacerbation and further away, we can see a statistically significant increase. So the take-home message from this is that the pathogens are behaving differently in different contexts. And this means for treatment that it might be relevant for the medical doctor to know the context in order to make a better targeted treatment, right? Because if I remove the pathogens from the anaerobes, likely nothing will happen, right? You will still have the clinical positive result that the pathogen is there, but if you treat it, nothing will happen. However, if you have a dysbiosis where the pathogen is driving, okay, then you can remove it, and this might, be a successful treatment. Okay, I don't know the time. Okay, perfect. Okay, so I hope that I could show you by analyzing this time series, very highly sampled from patients. We were looking at the organismic level, but also at changes in the community. And we found two different big regimes, even though we had these 3 clusters. The first one is this pathogen-dominated dysbiosis. The organization is a steep hierarchy. It has a reduced diversity and also overall simpler communities. And the turnover is stuck. And in this case, the CF pathogens act as master driver and they drive the dynamics. On the other hand, we have the anaerobic dominated regime. It could be some anaerobic flaring, some niches going out, is opening up, inviting more anaerobes coming in. The community organization is rather flat, it seems, and we have a high diversity, stochastic dynamics going on. And I think the most important part is, in this case, Prevotella and Streptococcus are in the major driving seat, however strong this driving seat effectively is. But I think the major importance is that the CF pathogens lack relevance in the dynamics of this regime. Yeah? And having said this, I think it is, it could be perhaps a pivotal knowledge if you're treating as a medical doctor to know about the community organization of your microbiome. Is this a dysbiotic situation or is this not a dysbiotic situation? Because it could be really be crucial for the intervention success. So having said this, and of course, there are many people to acknowledge. First, John Lipuma, who was the visionary who's actually collecting all these samples. He's at the University of Michigan. And then Chris Opron, who also helped me a little bit with coding, and my postdoc Lenka, and people and funding agencies, of course. And thanks for your attention, and I'm happy to take questions.
Thanks, Stefanie. Right on time. Questions, Caroline?
That sounds really interesting, but I have a doubt if it's, no, I don't have, I guess they are biologically meaningful, the clusters, but I have a doubt on the possibility of having the correct betweenness candidates from inferred networks, because we did an evaluation on that. Okay, the evaluation can be criticized as well, so maybe not very informative, but we could not find back with network inference tools betweenness, high betweenness nodes that we had put into the networks before. So I highly doubt that we are actually able to get the correct nodes.
Okay, I can repeat the question as far as I understood it. So it was a comment rather than a question. So in some validation on co-occurrence inference, I don't know the paper. So you put in some high between nodes, and you couldn't see them in the inference. Yeah. I would like to say something to this. Yeah, in what you're saying, you try to find something in a real interaction network, right? And you try to find it in the co-occurrence network. This is not my main objective, so to say. Yeah, I'm looking at the co-occurrence. This is a mixture of everything. We all know the limitations of co-occurrence, so we're well aware of this. Yeah, and still, I find the graph topology that I can somehow, clarify or I can define. And in that sense, I find betweenness nodes, which doesn't necessarily mean that the interaction network below that would reflect that betweenness node, right? But in terms of the overall co-occurrence, I do see them, right? So I wouldn't say that the organisms that are highly in between in a co-occurrence network are really in the interactions highly in between, but overall from the dynamics that you get and that you actually extract from the correlations, right? They are there. It's a way of, but we can discuss. I would be happy to discuss about this in more detail.
If I understood correctly, the samples here, these were all at points leading up to one of these exacerbations.
Yes, exactly.
I guess I'm confused to know how these different clusters of the dynamics actually correspond to differences in the patient outcomes. Like when they have the pulmonary
you mean whether there are more pronounced exacerbations, exactly, for example. I would love to know that, but I don't, because I don't have the exact metadata for this. Yeah, let's put it like this. So there is a query that is people. So these are very committed patients. Yeah, you have to imagine that these are daily sampled, a collection of daily samples. And this subset that we're using was really just a subset. So there are persons who over 2 years took daily samples. Yeah, and so of course when they started the collection and the study, they asked them also to fill in some questionnaires, but the questionnaires changed. and so in reality the questionnaires are useless, and so with regards to the real outcome in this individual exacerbation, so normally you would take lung function tests. There is a particular parameter that is very well used, FEV1 it's called, but unfortunately it's not so over the time when the patient is not coming to the hospital, there are no FEV1s, and then for the exacerbation themselves there are some are not there, so sporadic. So I would love to make this conclusion, but I cannot. Yeah.
Okay, maybe I can abuse my position and ask a related question. So especially with this anaerobic switch, are there any changes in the samples? Because people talk about formation of like structures in these systems sometimes. Are the samples getting like more granular or something like this?
No, so for me, I don't see so many switches, as I've been showing in the beginning. So I have the strong feeling that a patient having a dysbiosis keeps on having this dysbiosis despite various rounds of exacerbations and treatment. Yeah. It just comes back in a certain manner, right? So if you have a very strong Pseudomonas also in abundance and you treat it, and you treat everything out, then of course, like parts of all these organisms will survive, right? But you will have relatively more Pseudomonas to start with, and so it seems to me it's just, you know, rebooting, yeah.
A question. You mentioned this network analysis by Barabashi and Oldby, and then you interpret it as hierarchical network. But I think in the original interpretation of Barabashi, they called it scale-free network.
No, the degree distribution is scale-free, and with the clustering distribution, which also has to fit the power law, then you can interpret this is a hierarchical graph. Not the
interpretation. I don't think he spoke about that hierarchy.
No, it is from this. It is from this paper, or even the little logo that I had on top of this.
All right. If there are no questions, thanks, Stefanie, again. Okay, the next two talks are both contributed, and they both gonna be online. So I'm not sure if we can get them onto the screen here. All right.